Welcome to our channel. We are so glad you found your way here. Let us help you fall asleep. Let's begin with virus evolution, the non-living entities that shape life. Life allein sog lufte, faste core, take a slow, deep breath in and release it. Let the weight of the day fade away as we embark on an unhurried, atmospheric journey through the wonders of the cosmos. Your mind is quiet, the universe is vast, and we are ready to explore. Viruses occupy a unique space within biological sciences, challenging the very definitions of life. These structures, composed of genetic material encased in a protein coat, exhibit essential characteristics of living systems, yet lack the capacity for independent reproduction. Instead, they rely on host cells to replicate, blurring the lines between living and non-living. The diversity of viral forms is staggering, with each type exhibiting specific adaptations that allow them to infect a variety of hosts, ranging from bacteria to humans. This adaptability contributes to their evolutionary success, resulting in a vast array of viral genomes that can rapidly mutate and recombine. Such processes enable viruses to evade host immune responses, exemplifying the evolutionary arms race that exists within ecosystems. In the absence of metabolic processes, their existence is defined primarily by interactions with living organisms. This relationship illustrates a complex interplay of parasitism and coevolution, where viruses can influence host evolution and vice versa. The presence of these non-living entities has significant implications for understanding biological diversity and the mechanisms of disease. Viruses serve as agents of change in ecosystems, shaping not only their hosts but also the microbial communities that inhabit various environments. Their impact extends beyond immediate health concerns, influencing ecological dynamics and evolutionary trajectories across the biosphere. The exploration of viruses began in the late 19th century, marking a significant shift in scientific understanding. Initial observations revealed these entities could pass through filters designed to trap bacteria, suggesting a scale and complexity previously unrecognized. This region of inquiry, focusing on what would later be termed viruses, prompted researchers to reconsider the nature of life itself. The discovery of the tobacco mosaic virus in 1983 stood as a pivotal moment, illustrating that these non-living entities could possess genetic material and exhibit behaviors akin to living organisms. By the early 20th century, advances in microscopy allowed for greater clarity in visualizing these microscopic agents. Techniques developed during this period enabled scientists to observe the structural diversity of viruses, including their shapes and the arrangement of proteins. It became clear that they could adapt and evolve rapidly, a characteristic that would later play a crucial role in their interactions with hosts. The understanding of viral mechanisms progressed with the advent of molecular biology, revealing intricate processes like replication and mutation. These revelations contributed to the broader understanding of genetics and heredity, intertwining the fields of virology and evolutionary biology. Historical perspectives on virus discovery showcase an evolving narrative, illustrating the complexity of life and the myriad ways these entities influence both health and ecological systems. The intricate structure of viruses reveals a remarkable simplicity juxtaposed with profound complexity. Each virus is enveloped in a protective shell known as a capsid, composed of protein subunits called capsomers. This assembly not only shields the genetic material contained within, but also plays a crucial role in the virus's ability to infect host cells. The geometric arrangement of these proteins allows for a variety of shapes, ranging from helical to icosahedral forms, tailored for specific interactions with host organisms. Inside this protective casing lies the core of viral identity, the genetic material, which can be either deoxyribonucleic acid or ribonucleic acid. This genetic blueprint encodes the instructions necessary for replication and the synthesis of new virions. The arrangement of nucleotides within this material determines the characteristics of the virus, influencing its pathogenicity and transmission dynamics. Some viruses possess additional lipid envelopes derived from the host cell membrane, further enhancing their ability to merge with host cells. These structural features dictate not only the life cycle of the virus, but also its interactions with the immune system. Understanding these components provides essential insights into the mechanisms through which viruses operate, revealing their role as both agents of disease and fundamental players in the evolution of life on Earth. The intricate relationship between viruses and their hosts has profound implications for evolutionary biology. These entities, though non-living in the traditional sense, exert significant evolutionary pressure on various organisms. Their rapid mutation rates allow for swift adaptation, enabling them to evade the immune responses of hosts. In turn, this constant threat drives the evolution of complex immune systems, fostering diversity within species. Viruses also contribute to horizontal gene transfer, a process where genetic material is exchanged between different species. This mechanism can introduce new genetic traits into a host population, enhancing adaptability and survival in changing environments. Some essential genes in higher organisms are believed to have originated from viral ancestors, illustrating the intertwined nature of life's evolutionary pathways. In ecosystems, the presence of viruses can regulate population dynamics, influencing which species thrive and which decline. This regulation helps maintain ecological balance, demonstrating their role beyond mere pathogens. The evolutionary arms race between viruses and hosts exemplifies a continuous cycle of adaptation and counter-adaptation, shaping biodiversity over millions of years. This dynamic interaction showcases the dual nature of viruses as both harmful agents and catalysts for innovation in the living world. The study of these relationships enhances our understanding of life's complexity and the intricate web of connections that sustain it. Causification serves as a fundamental framework to categorize these non-living entities, underscoring their diverse structures and genetic materials. Among them, RNA viruses exhibit a remarkable ability to mutate quickly, allowing them to adapt to changing environments. Their genomes consist of ribonucleic acid, which can take various forms, including single-stranded and double-stranded configurations. This flexibility contributes to their rapid evolution and capacity to jump between hosts. In contrast, DNA viruses are characterized by their use of deoxyribonucleic acid as their genetic material. These entities often exhibit more stability in their genetic codes, resulting in lower mutation rates compared to their RNA counterparts. The classification of these viruses extends beyond mere genetic materials, encompassing various shapes, sizes, and modes of replication. Own's category plays a unique role in ecosystems, influencing everything from microbial populations to higher organisms. Understanding the distinctions between RNA and DNA viruses is crucial for developing effective vaccines and treatments. Their presence and interactions with living cells not only impact human health but also shape broader ecological dynamics. The ongoing study of virus classification reveals the complex interplay between these entities and life itself, highlighting their significance in the evolutionary narrative of all living organisms. The genetic code of viruses showcases a remarkable mosaic of life, composed of either deoxyribonucleic acid or ribonucleic acid. These molecular structures encapsulate the instructions necessary for viral replication and infection. Unlike cellular organisms, they exhibit an extraordinary variety of genetic configurations, ranging from single-stranded to double-stranded forms. This diversity allows them to adapt rapidly to changing environments, evading the defenses of their hosts. Ribonucleic acid viruses, in particular, display a high mutation rate. This characteristic fosters rapid evolution, enabling them to exploit new niches and host species. The implications of this adaptability are profound, influencing not only the health of individual organisms, but also entire ecosystems. Pathogenic viruses can alter ecological balances, leading to cascading effects throughout food webs. Genetic recombination also plays a crucial role in viral evolution. Through this process, segments of genetic material can be exchanged between different viral strains, resulting in novel variants with unique attributes. Such mechanisms have been observed in several significant viral outbreaks throughout history, underscoring the potential for sudden shifts in virulence and transmissibility. Understanding the genetic architecture of viruses provides insights into their roles within larger biological systems. This knowledge informs public health strategies and guides research aimed at countering viral threats, illustrating the intricate connections between these non-living entities and the living world. The origin of viruses remains a complex puzzle, with several hypotheses attempting to explain their emergence. One prominent theory suggests that they evolved from mobile genetic elements such as plasmids or transposons, which gained the ability to infect host cells. This mechanism proposes that they once served a role within cellular organisms before acquiring the capacity to hijack the machinery of living cells for replication. An alternative hypothesis posits that viruses emerged from the remnants of cellular life. This idea suggests that they may have descended from degenerate forms of once independent organisms, losing the essential functions needed for survival outside their hosts. Such a pathway indicates a gradual transition from complexity to simplicity, a lapse into a parasitic existence. Another consideration involves the possibility that viruses originated simultaneously alongside cellular life as a separate branch on the tree of life. This perspective presents them as ancient entities that have co-evolved with their hosts, adapting and diversifying over billions of years. Each of these theories contributes to the understanding of viruses as dynamic agents of evolution, capable of influencing genetic diversity and ecological balance. Exploration into the origins of viruses continues to shed light on their role in shaping life, revealing a realm where the boundaries between living and non-living entities are profoundly blurred. The evolutionary tree of life illustrates the intricate relationships among various organisms, positioning viruses in a unique context relative to cellular life forms. This structure, branching out through billions of years, captures the essence of evolutionary processes. with viruses often appearing as elusive branches that defy conventional categories of life. These entities, while not classified as cellular organisms, engage in constant interaction with hosts, contributing to genetic exchange and adaptation. Viruses can be viewed as evolutionary catalysts, exerting pressure on cellular organisms that drives evolutionary innovation. They influence host genome dynamics through mechanisms such as horizontal gene transfer, which allows for the acquisition of new traits and adaptations. This process highlights the interconnectedness of life, where even non-living entities play pivotal roles in shaping evolutionary trajectories. Some viruses exhibit relationships that border on symbiosis, integrating their genetic material into the genomes of host organisms, leading to functional advantages for both parties. These interactions illustrate the complexity of life's evolutionary history, Ocean Paulit, underscoring the importance of viruses as agents of genetic variability. In this context, the study of viruses enriches our understanding of evolutionary biology, revealing a landscape where life continuously adapts, reshapes, and evolves, influenced by the very entities once considered mere pathogens. The significance of these non-living agents continues to expand, inviting further exploration into their profound impact on the living world. Virus-host interactions exemplify a complex co-evolutionary dynamic where both entities adapt in response to one another. This relationship is characterized by an ongoing evolutionary arms race, with viruses developing strategies to evade host defenses, while hosts simultaneously evolve mechanisms to detect and eliminate these invaders. Such interactions are not merely adversarial, they can also drive significant innovations within host species, influencing genetic diversity and resilience. In this intricate network, the role of horizontal gene transfer becomes apparent. As viruses transfer genetic material between organisms, they can confer new traits that may enhance survival. This genetic exchange shapes evolutionary trajectories, giving rise to novel functions and adaptations. The interplay of viruses with their hosts may lead to the emergence of new species entirely, redefining ecological niches, and stances of this co-evolution can be observed in various ecosystems in aquatic environments. For example, phytoplankton have developed defenses against specific viral infections. Concurrently, viruses have evolved mechanisms to target and exploit these defenses, enhancing their infectivity. Such cycles of adaptation reflect a continuous evolutionary dialogue, where the success of one often hinges on the failure of the other. Through these interactions, the impact of viruses extends beyond mere infection, influencing ecological balance and evolutionary history across diverse life forms. This complexity underscores the significance of viruses as key players in the tapestry of life. Mutations serve as the primary mechanism driving the evolution of viruses, allowing for adaptation and survival in diverse environments. These alterations in the genetic material can occur spontaneously or as a response to external pressures, such as host immune defenses or antiviral treatments. Each mutation presents an opportunity for the virus to exhibit altered characteristics, which can significantly affect its transmissibility and virulence. The rapid replication rates of viruses contribute to a high mutation frequency. This phenomenon allows for the emergence of variants that may escape recognition by the immune system, thereby enhancing the virus's ability to infect hosts repeatedly. Environments rich in susceptible populations facilitate this process, leading to increased genetic diversity among viral strains. Some mutations confer advantageous traits that foster improved survival in specific contexts, while others may be neutral or deleterious. The interplay of these mutations shapes the evolutionary trajectory of viral lineages, culminating in a dynamic landscape of genetic variation. In the context of global health, the emergence of novel viral strains can create significant challenges for disease control, as seen with recent outbreaks. Understanding the mechanisms and consequences of viral mutations is essential for anticipating changes in pathogen behavior. This exchange occurs through horizontal gene transfer, a critical process in microbial evolution. Viruses act as vectors, facilitating the transfer of genetic material between organisms that would not typically share DNA. By injecting their genetic payload into host cells, they can inadvertently incorporate fragments of the host's genome into their own. This capability allows for the dissemination of advantageous traits across different species, potentially enhancing their survival and adaptability. In this region of genetic exchange, the impact of viral activity is profound. Bacterial populations, for instance, can acquire resistance genes through such interactions, rendering them impervious to specific treatments. This phenomenon alters the dynamics of microbial ecosystems, fostering resilience and rapid adaptation among populations. The implications extend beyond bacteria. They influence larger organisms as well. Eukaryotic cells can also experience genetic variation through viral-mediated transfer, contributing to evolutionary processes in complex life forms. This interaction supports the hypothesis that viruses, often perceived solely as pathogens, play a vital role in shaping genetic diversity. These dynamics underscore a duality in virology. While viruses can cause disease, they simultaneously serve as agents of change within biological systems. This intricate balance highlights the importance of studying viral mechanisms to grasp the broader implications for life on Earth and beyond. Viral reassortment stands as a pivotal process in the evolution of viruses, where genetic material is exchanged among different strains. This phenomenon occurs predominantly in segmented viruses, allowing for the combination of various genetic traits. Through reassortment, it is possible for a virus to acquire advantageous mutations, enhancing its adaptability and pathogenicity. In environments where multiple strains coexist, this exchange can lead to the emergence of novel viral variants. Such variants may possess enhanced abilities to evade host immune responses or exhibit increased virulence. This capacity for rapid adaptation underscores the dynamic nature of viral evolution. Each reassortment event can produce a variant with an entirely different profile, influencing not only the virus's behavior but also its interaction with host organisms. The consequences of viral reassortment extend beyond individual pathogens. It can reshape entire ecosystems, leading to shifts in the health and balance of populations. Infectious diseases may become more difficult to control, complicating public health responses. Understanding the mechanisms behind reassortment provides insight into the evolutionary pressures that drive the emergence of new strains, highlighting the interconnectedness of life forms in the face of non-living entities. This complex interplay emphasizes the necessity for ongoing research, illuminating the potential ramifications for both health and ecological stability. Quasar species represent a crucial element within viral populations, highlighting the dynamic and adaptive nature of these non-living entities. In contrast to traditional species concepts, quasi-species consist of a cloud of closely related genetic variants, all coexisting within a single viral population. This inherent variability allows for rapid evolution, enabling viruses to swiftly adjust to environmental pressures, such as host immune responses and antiviral therapies. Genetic drift plays a significant role in shaping these quasi-species, fostering diversity that can be advantageous in overcoming challenges. Through mutations, viruses can create strains that exhibit differences in virulence, transmissibility, and resistance to treatment. The interplay between quasi-species and their environments underscores the necessity of considering not only individual viral strains, but the collective behavior of these populations. The implications of this variability extend beyond mere survival. In the context of disease outbreaks, the presence of quasi-species can complicate vaccine development, as the evolving genetic landscape may outpace efforts to create effective immunizations. Understanding this phenomenon aids researchers in predicting potential trajectories of viral evolution. Recognizing the significance of quasi-species enriches our comprehension of viral behavior and their interactions with hosts, allowing for a deeper exploration into the mechanisms that govern the delicate balance of life and disease. The evolution of virulence reflects a complex interplay between viruses and their hosts, revealing how these non-living entities adapt to optimize their survival. Pathogenicity arises as a result of various evolutionary pressures, including host immune responses and environmental factors. This relationship is not static. It evolves as both the virus and the host engage in a continuous cycle of adaptation. A virus's ability to inflict damage on its host is not solely a reflection of its inherent properties, but also the host's defenses. In some instances, a more virulent pathogen may emerge when the host population has diminished immunity, allowing the virus to thrive. Conversely, reduced virulence can occur when a pathogen benefits from maintaining a balanced relationship with its host, ensuring prolonged transmission. The mechanisms underlying this evolution involve genetic mutations, recombination, and reassortment, which contribute to the diversity of viral strains. These processes can result in novel pathogenic traits, altering the effectiveness of host defenses. The study of viral evolution provides insights into the potential emergence of new pathogens, influencing public health strategies and vaccine development. The dynamics of virulence are further complicated by ecological changes, which can shift the balance between pathogens and hosts. Understanding these interactions is crucial for anticipating future outbreaks and managing the complex landscape of infectious diseases. The arms race between host immune responses and viral evasion strategies exemplifies a dynamic interplay that shapes the evolutionary trajectory of both entities. Immune systems, equipped with a multitude of defense mechanisms, continuously adapt to recognize and neutralize pathogens. These intricate responses include the production of antibodies, activation of T cells, and the release of signaling molecules, all orchestrating a comprehensive defense against invaders. And the resolve, in response, viruses employ an array of sophisticated strategies to circumvent detection and destruction. Genetic mutations can alter surface proteins, allowing them to evade host antibodies. Some viruses produce proteins that inhibit key immune pathways, effectively dampening the host's ability to mount a robust response. This ongoing cycle of adaptation fuels the relentless evolution of both sides, leading to an ever-changing landscape of infection and immunity. This region of interaction is not merely a battle, it is a complex evolutionary forge where the survival of the fittest takes place. Each viral adaptation can provoke a corresponding modification in host defenses, leading to a perpetual push and pull. The consequences of these interactions extend beyond individual hosts, shaping population dynamics and influencing the emergence of new viral strains. These processes underscore the intricate relationship between life and the non-living entities that inhabit it, revealing a profound complexity inherent in the fabric of existence. Environmental changes serve as a catalyst for viral evolution, influencing the characteristics and behavior of these non-living entities. Shifts in temperature, humidity, and habitat can create new challenges for viruses, prompting adaptations that enhance their survivability and transmission. In these dynamic conditions, mutations occur, leading to variations that may better suit the altered environments. Earth's ecosystems under stress often become fertile ground for the emergence of novel viral strains. This phenomenon is particularly evident in regions where climate change alters the distribution of host species. The movement of animals into new territories can introduce viruses to populations that have not previously encountered them, facilitating cross-species transmission. As these interactions unfold, viruses may undergo genetic recombination, further diversifying their traits. Such evolutionary processes can lead to outbreaks, as newly adapted viruses exploit vulnerabilities in host defenses. This cycle exemplifies the ongoing interplay between life and its surrounding environment, where each change ripples through the biological landscape, fostering new evolutionary pathways. Understanding the role of environmental changes in viral evolution sheds light on the broader implications for public health and ecological stability. Monitoring these shifts allows for better preparedness in anticipating potential viral outbreaks, emphasizing the need for a comprehensive approach to studying both viruses and their ecosystems. Rising global temperatures lead to shifts in ecosystems, creating favorable conditions for certain viruses to thrive. This region, once too harsh for many pathogens, may now provide a nurturing environment. The altered climate influences host species distribution, extending the geographical reach of viruses as their vectors adapt to new climates. Changes in precipitation patterns further affect the habitats of these organisms, leading to unpredictable viral outbreaks. As temperatures rise, the metabolic rates of both viruses and their hosts increase, resulting in altered interactions. The dynamics of transmission can shift dramatically, with some viruses becoming more virulent as they adapt to changing environmental conditions. In aquatic environments, warming waters facilitate the proliferation of waterborne viruses, impacting both wildlife and human populations. These transformations are not limited to terrestrial ecosystems. Arctic regions, where permafrost thaws, expose ancient viruses that have remained dormant for millennia. The potential for these re-emerging pathogens to influence contemporary ecosystems poses significant risks. The interconnectedness of climate change and viral dynamics highlights the fragility of ecological balance. Understanding these relationships is crucial in predicting future viral behavior, particularly as human activities continue to exert pressure on natural systems. This evolving landscape demands a rigorous scientific approach, combining virology, ecology, and climate science to mitigate potential health risks. Zoonotic viruses emerge from the intricate interplay between wildlife and human populations. These pathogens transition from animal reservoirs to humans, often during moments of ecological disruption. Spillover events occur frequently in regions where habitats are encroached upon, leading to increased interactions between species. This region witnesses heightened opportunities for pathogens to leap from one host to another, facilitated by environmental changes such as deforestation or urban expansion. Once in a new host, the viruses undergo a process of adaptation, evolving to exploit the biological nuances of their new environment. Genetic mutations enable these agents to enhance their infectivity and transmission capabilities. This adaptability is often propelled by the diverse genetic backgrounds of potential hosts, creating a dynamic landscape for viral evolution. The consequences can be significant, as demonstrated by recent outbreaks. These incidents underscore the importance of monitoring wildlife health, as well as understanding the ecological factors that contribute to spillover. Surveillance efforts focus on identifying potential risks in wildlife populations, providing essential data for predicting future outbreaks. Recognizing the interconnectedness of ecosystems is crucial. The delicate balance between human activity and wildlife habitats plays a pivotal role in shaping the emergence of zoonotic viruses. Vigilance and responsibility are paramount in addressing the challenges posed by these evolving biological entities. The intricate interplay between viruses and their environments reveals a complex ecological niche. These non-living entities inhabit diverse ecosystems, from soil to the human body, each uniquely influencing the balance of life. In aquatic environments, they play a crucial role in regulating bacterial populations, effectively maintaining the health of marine ecosystems. This regulation prevents overgrowth, ensuring nutrient cycles function smoothly. In terrestrial habitats, their presence impacts plant health, influencing agricultural systems and natural vegetation. Specific viruses can enhance the resilience of crops against pests, while others may lead to significant crop losses. The continuous interaction between viruses and hosts is a driving force in evolutionary processes, fostering genetic diversity within populations. Symbiotic relationships have been observed, where certain viruses contribute to the fitness of their hosts. These interactions challenge traditional views on pathogenicity, highlighting the dual nature of viruses as potential facilitators of ecological balance. Through horizontal gene transfer, they act as vectors of genetic information, promoting adaptation and evolution across various life forms. Understanding this ecological role is essential for comprehending the broader implications of viral evolution. The influence of these entities extends beyond immediate health concerns, intertwining with the fabric of ecosystems and shaping the evolutionary trajectories of countless organisms. This nuanced perspective fosters a deeper awareness of their significance in life's intricate web. Bacteriophages, the viruses that infect bacteria, play a pivotal role in shaping microbial populations within diverse ecosystems. By selectively targeting specific bacterial hosts, these entities regulate the abundance and diversity of bacterial communities. This interaction not only influences the dynamics of microbial life, but also has cascading effects on nutrient cycling and the overall health of environments. In aquatic ecosystems, bacteriophages contribute to the turnover of bacterial biomass, promoting the rapid recycling of organic matter. Their presence can lead to the suppression of harmful bacterial strains, thereby fostering a more balanced microbial landscape. The intricate relationships between these viruses and their bacterial hosts can be viewed as a delicate equilibrium, where fluctuations in phage abundance can trigger significant shifts in microbial composition. Research indicates that bacteriophages can enhance bacterial evolution through mechanisms such as horizontal gene transfer, providing genetic material that may confer new survival advantages. This evolutionary pressure can drive the emergence of antibiotic resistance, presenting both challenges and opportunities for medical science. In agricultural settings, The application of bacteriophages offers a potential strategy for controlling pathogenic bacteria without disrupting beneficial microbial populations. Such interventions illustrate the profound implications of these non-living entities as they navigate through the complex web of life, influencing health and sustainability in varied ecosystems. The interactions between plant viruses and their hosts reveal intricate evolutionary strategies that have developed over millennia. They exploit vulnerabilities within the cellular machinery of flora, often manipulating the host's defenses to ensure their replication and spread. This region of study uncovers a remarkable resilience among plants, which have evolved sophisticated mechanisms to combat viral infections. Unangdind oftazay. For reasons, viruses adapt, constantly refining their strategies to bypass these defenses. This ongoing evolutionary arms race shapes the genetic landscape of both the viruses and their hosts. Specific plant species may develop resistance traits, while the viruses simultaneously mutate, leading to new strains that can overcome previous barriers. Such dynamics contribute to the diversity of flora observed in various ecosystems. Specific viral infections can facilitate horizontal gene transfer, allowing beneficial traits to spread within and between plant populations. Consequently, this interaction can enhance the adaptability of plants to changing environmental conditions, influencing agricultural productivity and biodiversity. The presence of viruses also plays a role in ecosystem dynamics, affecting plant community structures and nutrient cycling. By altering growth patterns and survival rates, these entities impact food webs and interactions among different species. Understanding these relationships is crucial for managing crops and conserving plant diversity, highlighting the essential role that viruses play in shaping the evolutionary trajectory of flora. Fungal viruses represent a largely overlooked facet of virus evolution, possessing unique characteristics that distinguish them from their more widely studied plant and animal counterparts. These entities, often residing within fungal hosts, can influence host behavior and ecology in profound ways. Their presence may alter growth patterns, reproductive strategies, and even competitive dynamics among fungal populations, contributing to the broader ecosystem's health and stability. In this realm, the interactions between viruses and fungi extend beyond mere infection. Some fungal viruses can induce beneficial traits in their hosts, enhancing resistance to environmental stresses or pathogenic organisms. These adaptations underscore the complex web of relationships that define life, illustrating that viruses are not merely agents of disease, but can also be agents of change. The study of fungal viruses reveals a diversity of genetic material, with many exhibiting distinct replication strategies that challenge traditional categorizations. They often rely on their hosts for replication, integrating into the fungal genome and influencing gene expression. This intricate relationship facilitates an evolutionary arms race, where both parties continually adapt to each other's strategies. As research advances, the implications of fungal viruses on agriculture and biodiversity become more pronounced. Recognizing their potential impacts could inform management practices, fostering a deeper understanding of how these non-living entities shape life on Earth. The evolution of viral entry mechanisms illustrates a complex interplay between viruses and their hosts. These entities have developed sophisticated strategies to infiltrate host cells, adapting over time to circumvent immune defenses and exploit cellular machinery. Proteins on their surfaces often mimic host molecules, enabling them to gain access without raising immediate alarms. In this region of viral biology, The variation among entry mechanisms is remarkable. Some viruses utilize receptor-mediated endocytosis, a process where the host cell engulfs the virus, allowing it entry. Others employ a direct fusion method, merging their membranes with the host, facilitating immediate release of genetic material into the cytoplasm. These adaptations are not static. They reflect an ongoing evolutionary arms race. Mutations in viral genomes can lead to altered entry strategies, enhancing their ability to infect a broader range of hosts or evade specific immune responses. This constant evolution shapes the landscape of viral prevalence and pathogenicity, influencing outbreaks and public health responses. The implications for vaccine development and antiviral therapies are significant. Understanding the mechanics of entry can guide targeted interventions. Viral replication encompasses several distinct phases that contribute to its life cycle. Initially, attachment occurs, where a virus binds to specific receptors on the surface of a host cell. This critical interaction can dictate host range and tissue tropism, influencing which organisms or cell types the virus can infect. Following attachment, penetration into the host cell takes place, allowing the viral genome to enter the cellular environment. Once inside, uncoating occurs, during which the viral capsid disassembles, releasing the genetic material into the host's cytoplasm. This marks the transition to the replication phase. The viral genome, whether DNA or RNA, hijacks the host's cellular machinery, directing it to synthesize viral proteins and replicate its genetic material. This phase is vital as it determines the quantity of viral components produced. Assembly follows, where newly formed viral particles are assembled within the host cell. This process can be influenced by factors such as cell type and the availability of cellular resources. Finally, the release phase occurs. Viruses exit the host cell, often causing cellular damage or death. This stage can involve lysis, where the cell bursts, or budding, a more subtle process that allows for continued host viability. Each phase plays a crucial role in the virus's ability to propagate and evolve within ecosystems, influencing the dynamics of infectious diseases. Viral enzymes serve as essential agents in the evolutionary adaptation of these non-living entities. They facilitate various biochemical reactions, enabling viruses to replicate and modify their genetic material. This characteristic plays a pivotal role in their adaptability, influencing how these pathogens interact with host organisms. The presence of polymerases, for instance, allows for the rapid synthesis of viral genomes, creating opportunities for mutations. These alterations can lead to enhanced infectivity or resistance against the host's immune responses. In this context, the rate of mutation becomes critical. High mutation rates can result in a diverse pool of viral variants, some of which may possess advantageous traits. This genetic variability is a cornerstone of viral evolution, allowing them to exploit new ecological niches or overcome host defenses. Within the confines of a single infection, a multitude of variants may emerge, each vying for dominance. The genetic landscape of a host organism significantly influences viral evolution. Variability in host immunity can create environments that favor certain viral mutations, allowing them to thrive. This interaction often leads to the emergence of viral strains that are better adapted to evade the host's immune responses, resulting in a subtle arms race between the virus and its host. The complexity of human genetic diversity plays a crucial role in shaping these interactions. Different populations exhibit varying susceptibility to infections, which can drive the evolutionary trajectory of viruses. Instance, the presence of specific alleles in the human genome can alter immune responses, thereby influencing which viral variants are more likely to succeed in a given population. Environmental factors also contribute to this dynamic. Geographic isolation can lead to distinct evolutionary paths for viral strains, as they adapt to the unique genetic makeup of local host populations. The interplay between host genetics and environmental pressures creates a unique microcosm where viral evolution can be both rapid and unpredictable. Viral evolution is not merely a consequence of random mutations, but is intricately linked to the genetic characteristics of their hosts. Understanding these relationships is crucial for predicting how viruses may adapt in the future and for developing effective public health strategies to combat infections. Trade-offs in replication strategies play a significant role in virus evolution. Each entity must balance the need for rapid reproduction with the ability to survive in various environments. High replication rates can lead to increased mutations, which may confer advantages in adaptability, but can also result in detrimental errors. This delicate equilibrium influences not only the virus's longevity, but also its pathogenicity. Certain viruses have developed strategies that allow for slower replication, enabling them to maintain genetic stability over time. This approach can enhance their capacity to establish persistent infections, ensuring survival within host populations. In contrast, others prioritize rapid replication to outpace the immune responses of their hosts, leading to swift transmission. The environment, including host availability and immune system pressures, further complicates these trade-offs. Viruses often adapt their strategies based on ecological niches where they find themselves. For instance, in densely populated areas, rapid replication may be favored, while in isolated environments, a more conservative approach might prevail. These evolutionary strategies are not static. They evolve in response to changing conditions, including the emergence of antiviral treatments or vaccines. The intricate interplay of these factors shapes the landscape of viral evolution and highlights the adaptive capabilities of these non-living entities in the ecosystems they inhabit. Antigenic drift and shift are pivotal mechanisms in the evolution of influenza viruses, contributing to their remarkable adaptability. Antigenic drift occurs through the gradual accumulation of mutations in the viral genome, leading to small changes in the surface proteins known as antigens. These alterations can render previous immune responses less effective, facilitating the virus's continued circulation within populations. Over time, even minor genetic variations can significantly impact the virus's ability to evade host defenses. Antigenic shift represents a more drastic change, resulting from the reassortment of genetic material when two different influenza viruses infect the same host cell. This process can produce novel strains with entirely new antigenic profiles, creating the potential for pandemics. Such shifts may arise from interactions between human and avian strains of the virus, illustrating the interconnectedness of various species in viral evolution. The consequences of these mechanisms are evident in the seasonal patterns of influenza outbreaks. Each year, the virus exhibits variations that challenge the effectiveness of existing vaccines, necessitating continuous monitoring and adaptation by public health authorities. Understanding these processes is crucial for developing effective prevention strategies and treatments. This region of study underscores the dynamic nature of viral evolution and the ongoing arms race between pathogens and host immune systems, shaping the landscape of infectious diseases globally. Vaccine strategies have evolved significantly in response to the ever-changing threat of viruses. These approaches focus on anticipating viral mutations and adjusting formulations to ensure effectiveness. Traditional methods, which often relied on inactivated or attenuated viruses, laid the foundation for modern developments. The discovery of recombinant DNA technology enabled the production of safer and more targeted vaccines, paving the way for innovative solutions. Advancements in mRNA technology mark a pivotal shift in vaccine design. This method utilizes genetic instructions to prompt the body's cells to produce a harmless component of the virus, subsequently eliciting an immune response. This approach allows for rapid development and deployment, exemplified during the recent global pandemic. Swiftly adapting to viral mutations has become paramount, and ongoing research is crucial in refining these strategies. Combination vaccines that address multiple pathogens simultaneously are gaining traction, potentially enhancing public health responses. Such formulations aim to bolster immunity against a spectrum of viral threats, increasing resilience within populations. The emergence of nanoparticle-based vaccines offers another promising avenue, enabling precise targeting and enhanced immune activation. Ongoing surveillance of viral strains ensures that vaccine strategies remain aligned with the current landscape. Continuous innovation, driven by the interplay of scientific research and epidemiological data, shapes the future of viral prevention efforts. The development of pharmaceuticals introduces a new dynamic within viral ecosystems. Antiviral agents exert selective pressure on viruses, influencing their genetic makeup and fostering mutations. These alterations can lead to resistance, thereby complicating treatment protocols. In this environment, viruses adapt, evolving rapidly in response to the presence of these therapeutic agents. Targeted therapies, designed to inhibit specific viral functions, often find their efficacy diminished over time. The emergence of resistant strains illustrates a fundamental principle of evolutionary biology. Those that survive adapt. This phenomenon underscores the importance of continuous monitoring and innovation within pharmaceutical approaches. The role of vaccines also cannot be overlooked. Immunization not only protects individuals, but can shift the transmission dynamics within populations. This herd immunity establishes a barrier, limiting the spread of certain viral strains. Nonetheless, the potential for breakthrough infections highlights the need for ongoing vaccine development and adaptation to evolving viral threats. Research into combination therapies demonstrates a proactive strategy against resistance. By employing multiple pharmaceuticals simultaneously, the chances of viral escape are reduced. Such approaches exemplify the intricate relationship between human intervention and viral evolution. Each step taken in pharmaceutical development informs the ongoing dialogue with these non-living entities, revealing the complex interplay between humanity and the pathogens that challenge it. Globalization facilitates unprecedented movement among populations, allowing for the rapid dissemination of viruses across vast distances. This interconnectedness creates a landscape where pathogens can efficiently exploit opportunities for transmission. In this environment, viruses adapt and evolve, developing new strains that may exhibit enhanced virulence or resistance to existing treatments. The exchange of goods, travel, and communication fosters conditions that promote genetic diversification among viral populations. As people migrate, they bring with them a unique microbiome that interacts with local pathogens, potentially leading to the emergence of hybrid strains. These adaptations can render previous vaccines less effective, necessitating continual updates in immunization strategies. Various ecosystems, once isolated, now experience novel interactions as species migrate due to climate change and human activity. Viruses may leap from one host species to another, exploiting these new niches. The evolution of zoonotic viruses becomes particularly pronounced in areas where human encroachment alters natural habitats. Here, mutations may occur that enhance their ability to infect humans, posing new public health challenges. The consequences extend beyond immediate health concerns, influencing economic systems and societal structures. Understanding these evolutionary dynamics is crucial for developing robust strategies to mitigate the impact of viral outbreaks, emphasizing the need for comprehensive surveillance and response mechanisms in a globally interconnected world. Social behaviors significantly influence the pathways through which viruses spread. The interaction patterns among individuals create potential avenues for transmission, facilitating the movement of these non-living entities through populations. Close contact, communal activities, and shared environments enhance opportunities for viral particles to leap from host to host. This region, where social ties are strong, often becomes a breeding ground for outbreaks. Behavioral choices such as hand hygiene practices, mask wearing, and adherence to vaccination protocols play a critical role in determining the overall susceptibility of communities. The dynamics of social gatherings, from crowded public events to intimate family gatherings, dictate the likelihood of close encounters that can lead to viral dissemination. As individuals engage in daily routines, the interconnectedness of their lives can either amplify or attenuate the spread of pathogens. Cultural factors also contribute significantly to these behaviors, shaping perceptions regarding health and safety. Trust in public health measures can influence compliance, while misinformation may lead to neglect of precautionary practices. In this context, the societal response to a viral threat becomes a complex interplay of psychology, culture, and communication. Recognizing these social dimensions not only enhances the understanding of transmission dynamics but also underscores the importance of targeted public health interventions that resonate with community values and behaviors. Epidemiology plays a critical role in shaping viral evolution, influencing how these entities adapt and persist within populations. The study of disease patterns and their determinants provides insight into the selective pressures that viruses encounter. Environments with dense populations, for instance, foster rapid transmission, accelerating mutation rates as viruses adapt to new host defenses. In these regions, the interaction between humans and viruses can lead to the emergence of variants, resulting from genetic changes that provide an advantage in particular contexts. This evolutionary pressure is further intensified by vaccination efforts, which introduce additional factors into the viral landscape. The presence of vaccines can create niches where only certain viral strains survive, forcing them to evolve in response to immune pressures. Geographical variations also impact viral evolution, as distinct ecosystems present unique challenges and opportunities. These differences can lead to localized adaptations that may not be present in other environments. The movement of populations across borders can further complicate these dynamics, facilitating the spread of newly evolved strains into previously unaffected areas. Surveillance of viral evolution through epidemiological studies is essential for anticipating future outbreaks. The patterns observed can inform vaccine development and public health strategies, ensuring that responses remain effective in mitigating the impact of these evolving entities on human health. Human activity has significantly altered the landscape in which viruses evolve. Urbanization, deforestation, and climate change have created new ecological niches, allowing these entities to adapt and proliferate. The alteration of habitats often leads to increased interactions between humans and wildlife, facilitating the spillover of viruses from animals to people. This phenomenon underscores the interconnectedness of ecosystems and the role of anthropogenic influences in shaping viral evolution. In this shifting environment, selective pressures abound. Changes in temperature and rainfall patterns affect the distribution of both hosts and vectors, leading to shifts in viral circulation. The emergence of novel strains can often be traced back to these environmental transformations, highlighting the dynamic nature of viral genomes as they respond to human-induced changes. Surveillance mechanisms are vital for monitoring the emergence of these pathogens, Advances in genomic sequencing have allowed researchers to track mutations and understand the pathways through which viruses adapt. This ongoing research forms a crucial component of public health strategies, bridging the gap between environmental science and human health. The implications of these interactions extend beyond immediate health concerns. Understanding how human influence affects viral evolution can guide policy decisions aimed at reducing risk factors associated with zoonotic spillover. The role of bioinformatics in understanding virus evolution is pivotal. Advanced computational techniques allow researchers to analyze vast amounts of genetic data, revealing patterns that would remain obscured through traditional methods. This field integrates biology, computer science, and mathematics, facilitating the examination of viral genomes and their mutations. Through the application of algorithms, it becomes possible to track the evolution of viruses across different hosts and environments. Bioinformatics tools enable the reconstruction of phylogenetic trees, illustrating the relationships between various viral strains. By comparing genetic sequences, scientists can identify common ancestors and trace the lineage of specific viruses. This capability is crucial in understanding how viruses adapt to new environments and hosts, contributing to their evolutionary success. Data mining from clinical records and environmental samples enhances knowledge about viral outbreaks, patterns of transmission and mutation, offer insights into the mechanisms that drive these non-living entities. The integration of geographical information systems further helps to visualize the spread of viruses across different regions. Collaboration among researchers worldwide is essential for building comprehensive databases that house genetic information and epidemiological data. These efforts create a more complete picture of viral evolution. Phylogenetic studies serve as a pivotal tool in mapping virus evolution, establishing connections between various strains and elucidating their relationships over time. By analyzing genetic sequences, researchers can trace back the lineage of viruses, unveiling the alterations that occur through mutations and recombinations. This process reveals how environmental pressures and host interactions influence viral adaptation, shaping their survival strategies in a dynamic ecosystem. In this region of study, the use of molecular phylogenetics has become increasingly sophisticated. Techniques such as next-generation sequencing allow for the rapid decoding of viral genomes, providing insights into their evolutionary histories. The resulting phylogenetic trees visually represent these relationships, illustrating how closely related certain viruses are and how they diverged from common ancestors. These trees are not merely academic exercises. They have practical implications for understanding outbreaks. Identifying viral lineages helps predict transmission patterns, informing public health responses. The emergence of new strains can be tracked, allowing for timely interventions. Continued advancements in computational biology enhance the accuracy of these analyses, revealing intricate networks of viral evolution that span continents and decades. As researchers delve deeper into the genetic intricacies of these non-living entities, the understanding of their role in shaping life becomes increasingly precise, highlighting the delicate balance within ecosystems. The evolution of viral pathogenicity offers a distinct perspective on these entities and their impact on living organisms. Case studies reveal how certain viruses have adapted specific mechanisms to facilitate infection and replication within host cells. The process often begins with mutations that enhance their ability to bind to host receptors, allowing for a higher likelihood of transmission. In some instances, genetic reassortment occurs when two different viruses infect the same host cell, leading to the emergence of novel strains. This has been observed in influenza viruses, where shifts in surface proteins can result in pandemic outbreaks. Such adaptations illustrate the dynamic nature of viral evolution, emphasizing how quickly these entities can respond to environmental pressures. Another notable case study involves the human immunodeficiency virus, which demonstrates a remarkable ability to evade immune detection through rapid mutation. This characteristic not only complicates treatment strategies, but also underscores the ongoing arms race between pathogens and host defenses. The impact of these evolutionary changes extends beyond individual hosts, influencing the overall health of populations and ecosystems. The interplay between viral evolution and host responses provides insight into the broader implications of infectious diseases, underscoring the intricate relationships that define life on Earth. Understanding these processes contributes to the development of effective prevention and control measures in public health. The influence of host population structure on viral evolution is profound and multifaceted. Diverse host interactions create unique environments where viruses can adapt and thrive. Genetic diversity within populations provides a reservoir of potential targets for viral adaptation, enabling the emergence of new variants. This variation can occur as viruses exploit different cellular machinery across host species, or even within subpopulations of the same species, leading to differences in replication rates and transmission dynamics. Geographical distribution also plays a significant role. Isolated populations may experience unique viral pressures, resulting in distinct evolutionary trajectories. In densely populated urban areas, for instance, rapid transmission cycles foster the selection of more transmissible strains. In contrast, rural settings may permit more stable, less aggressive viral forms to persist. This dynamic can create a patchwork of viral strains, each adapted to its specific host context. Interactions among hosts influence viral evolution as well. Co-infections can lead to genetic recombination, producing hybrid viruses that may possess novel properties. The host immune response further shapes these evolutionary pathways. Different immune pressures can favor various viral adaptations, leading to a continuous arms race between host defenses and viral strategies. Viral co-infections introduce a complex layer to evolutionary dynamics. When multiple viruses invade a single host, they can interact in unpredictable ways, influencing both their own evolution and that of the host. These interactions can lead to increased genetic diversity among the viruses, as they may exchange genetic material through a process known as recombination. This genetic promiscuity can result in the emergence of novel viral strains, potentially evading existing immune responses. The presence of co-infections can alter the immune system's response, creating a scenario where the host's defenses are stretched thin. In some cases, the host may experience a dampened immune response, allowing less virulent strains to persist and spread more effectively. This phenomenon can lead to a paradoxical increase in overall viral load, as the immune system struggles to cope with the multitude of pathogens. These dynamics highlight the role of co-infections as a significant factor in shaping viral evolution. The interplay between different viral entities can accelerate adaptation, making it essential for researchers to consider these interactions when studying viral behavior and disease outbreaks. In environments rich with co-infecting agents, evolutionary trajectories can diverge rapidly, leading to unforeseen consequences for both the viruses involved and their hosts. Understanding this complexity is crucial in predicting future viral emergence and designing effective intervention strategies. The future of viral evolution in a changing world hinges on numerous interrelated factors, including climate change, urbanization, and global connectivity. These elements create new ecological niches, facilitating opportunities for viruses to adapt and thrive in altered environments. This region, once characterized by stable ecosystems, now experiences shifts that favor microbial survival and proliferation. Dynamic interactions among species will increasingly dictate viral evolution as host populations undergo stress from environmental changes. These changes may lead to genetic mutations, allowing certain strains to exploit new hosts or evade immune responses. The implications for public health are profound, with the potential emergence of novel pathogens that could challenge existing medical interventions. Global travel accelerates the spread of viruses, amplifying the risks associated with emerging infectious diseases. Each movement introduces pathogens to new populations, where they may encounter diverse genetic backgrounds. This interaction contributes to the ongoing evolutionary arms race between viruses and their hosts. Research efforts must adapt to these shifting paradigms, focusing on surveillance and rapid response capabilities. Understanding the mechanisms driving viral evolution will be essential for predicting potential outbreaks and mitigating their impact on human populations. In this evolving landscape, the study of viruses as non-living entities will play a pivotal role in shaping future strategies for global health security. Emerging viral diseases serve as a critical focal point in evolutionary studies, revealing the intricate relationships between pathogens and their hosts. These viruses adapt rapidly, often exploiting genetic variability within populations to survive. The continuous mutation and recombination processes among viral genomes facilitate this adaptability, enabling them to evade immune responses and develop resistance to antiviral therapies. In this context, the interaction between viruses and host organisms illuminates broader ecological dynamics. For instance, the spillover of viruses from animal reservoirs to humans underscores the importance of biodiversity in these environments. The health of ecosystems influences the emergence of new viral strains, indicating a delicate balance that must be preserved to mitigate future outbreaks. Genomic sequencing technologies have advanced significantly, allowing researchers to trace lineages and understand the evolutionary timelines of these pathogens. This data provides insights into how emerging viral diseases can affect human populations and influence evolutionary trajectories. Each outbreak offers a unique opportunity to study viral adaptation and the selective pressures faced by hosts, contributing to a more comprehensive understanding of evolutionary biology. The implications extend beyond mere viral characteristics. They encompass the socio-economic impacts of emerging diseases, emphasizing the need for interdisciplinary approaches in studying these phenomena. The interplay between viruses and their environments continues to reshape the evolutionary narrative of life on Earth. Synthetic biology has emerged as a powerful tool that enables the manipulation of biological systems, including viruses. Through engineering, it becomes possible to create novel viral entities or modify existing ones to better understand their interactions with host organisms. This manipulation can lead to significant insights into viral behavior, replication, and pathogenicity. The ability to design viruses with specific traits opens up unprecedented opportunities for research and medicine. By altering viral genomes, scientists can explore how these entities adapt to various environmental conditions and host defenses. This knowledge not only aids in the development of vaccines, but also enhances therapeutic strategies against viral infections. In this evolving landscape, the implications of engineered viruses extend beyond immediate medical applications. The deliberate introduction of synthetic viruses into ecosystems poses questions about ecological balance and evolutionary pressures. These modified entities may outcompete native viruses, altering the dynamics of viral populations and reshaping host interactions in unforeseen ways. The integration of synthetic biology with virology thus represents a double-edged sword. While it holds promise for advanced medical solutions, it also necessitates careful consideration of long-term consequences on evolution. Monitoring the outcomes of such interventions will be crucial in understanding how engineered viruses influence the broader tapestry of life, potentially driving new evolutionary pathways. An thain nichand vi weidmothum fiat forik kostiv. CRISPR technology represents a pivotal advancement in the ongoing struggle between organisms and viruses. This adaptive immune system, originally discovered in bacteria, has been harnessed to create precise genetic modifications across a wide range of species. It serves as a powerful tool for researchers seeking to understand the intricate dynamics of viral interactions. By targeting specific sequences within viral genomes, CRISPR can effectively disable these non-living entities, disrupting their ability to replicate and spread. The implications of this technology extend far beyond laboratory settings. In nature, the evolutionary arms race between hosts and viruses continues to drive innovation. Organisms develop new defenses in response to viral threats, while viruses rapidly evolve mechanisms to evade detection and counteract these defenses. This relentless cycle results in a complex interplay, shaping the genetic landscape of entire ecosystems. Advances gained from CRISPR applications illuminate how these organisms adapt in real time. Each genetic alteration creates potential new vulnerabilities, which viruses may exploit. Consequently, the relationship between these entities is not static. It is a dynamic and ever-evolving process. Observing these interactions contributes to a deeper understanding of evolution itself, revealing the intricate web of life shaped by both the persistence of viruses and the resilience of their hosts. The ongoing research into these mechanisms continues to offer critical insights into the foundations of biological diversity. Antiviral resistance emerges as a significant factor influencing the evolution of viruses. This phenomenon occurs when viral populations adapt in response to the selective pressure exerted by antiviral agents. The introduction of these treatments creates an environment where only those viral strains capable of surviving the drugs can propagate. These resistant strains often exhibit mutations that alter their genetic makeup, enabling them to thrive despite the presence of therapeutic interventions. The dynamics of this process are evident in various viral infections, where persistent exposure to antivirals leads to the emergence of new variants. These variants may demonstrate enhanced fitness in the absence of treatment, but may also carry trade-offs affecting their virulence or transmissibility. As a result, the landscape of viral populations continually shifts, responding to both human medical practices and ecological factors. Understanding the mechanisms behind antiviral resistance is crucial for developing effective treatment strategies. Genetic studies reveal the pathways through which viruses acquire resistance, highlighting the role of mutations and recombination events. This knowledge aids in predicting future trends in viral evolution, allowing researchers to anticipate challenges in managing infections. The implications of these evolving viruses extend beyond individual health, influencing public health policies and the development of vaccines. The interplay between antiviral resistance and virus evolution underscores the complexity of managing infectious diseases in a changing environment. The emergence of COVID-19 illustrates the dynamic nature of viral evolution. Originating from a zoonotic transmission, this virus adapted rapidly within human populations, showcasing the mechanisms of mutation and selection. Genetic analyses reveal that mutations allowed it to evade certain host immune responses, facilitating its spread. These alterations highlight the influence of evolutionary pressures exerted by human behavior, environmental factors, and global connectivity. Transmission routes were not limited to direct contact, but included aerosols, leading to widespread outbreaks. This mode of spread intensified the selection for variants that enhanced transmissibility. Observations of the virus's genetic drift demonstrate how minor changes can significantly impact its behavior and interaction with human immune systems. The emergence of variants of concern, such as Alpha, Beta, and Delta, exemplifies this phenomenon, each exhibiting unique characteristics that challenge public health responses. The ongoing evolutionary trajectory of COVID-19 raises questions about the future landscape of viral threats. Surveillance and genomic sequencing remain critical tools in monitoring these changes, offering insights into how emerging variants may shape the course of the pandemic. Understanding this evolution assists in vaccine development and the formulation of effective treatments, emphasizing the necessity of adaptability in public health strategies. This intricate process underlines the perpetual dialogue between virus and host, a dance of adaptation that continues to unfold. The microbiome, a complex ecosystem of microorganisms residing within various environments, is influenced significantly by these non-living entities. Viruses play a crucial role in shaping its composition and function. They infect bacteria, archaea, and even eukaryotic cells, driving a cycle of predation and competition that regulates microbial populations. This interaction ensures that no single species dominates, maintaining ecological balance within the microbiome. The presence of viruses can trigger genetic exchanges among microbial communities, a process known as horizontal gene transfer. This mechanism allows for the swift acquisition of new traits, including antibiotic resistance and metabolic capabilities. Such adaptations enhance the resilience of microbial populations in response to environmental pressures. In this vast and dynamic system, viruses act as both predators and facilitators. They selectively remove specific bacterial strains, enabling others to thrive. This selective pressure can reshape community structures, influencing health outcomes in hosts. The symbiosis between host organisms and their microbiomes is thus deeply affected by viral activity. Recent studies have begun to uncover the nuanced relationships within this microbial consortium, revealing how viruses can influence inflammation, immunity, and even behavior in their hosts. The intricate web of interactions highlights the delicate balance that governs life, emphasizing the importance of understanding these non-living entities in the broader context of biological systems. Oncolytic viruses, a unique subset of these non-living entities, have undergone significant evolution in their application to cancer therapy. This development embraces the inherent ability of certain viruses to selectively infect and destroy cancerous cells, while sparing healthy tissues. Research has revealed that the genetic modifications of these viruses enhance their specificity, allowing for targeted interventions that minimize collateral damage. And in the Wevernamedwainty settings, these engineered strains exhibit remarkable efficacy against various tumor types. The mechanisms of action involve direct oncolysis, where viral replication leads to the lysis of malignant cells, and indirect effects, such as the stimulation of anti-tumor immune responses. This dual approach amplifies the therapeutic potential, suggesting a synergistic relationship between the viral agents and the host's immune system. Clinical trials have demonstrated promising results, with patients experiencing reduced tumor burden and improved survival rates. The evolution of oncolytic viruses continues to be a focal point in oncology research, as scientists explore combinations with other therapeutic modalities. such as checkpoint inhibitors and chemotherapy. Each advancement opens new avenues for treatment, driven by a deeper comprehension of viral biology and cancer immunology. The implications of these findings extend beyond mere therapy, posing profound questions about the coevolution of viruses and host organisms, enriching our understanding of health and disease. This interplay between viruses and other pathogens reveals a complex web of interactions that can influence the dynamics of infectious diseases. Viruses often facilitate or inhibit the spread of bacteria and fungi, altering the landscape of microbial communities within hosts. Certain viral infections can lead to a phenomenon known as viral-induced bacterial lysis, where the presence of viruses causes bacterial cells to burst, releasing nutrients that subsequently may favor the growth of other microbial species. These microbial shifts can have significant ecological consequences. For instance, in environments where diverse pathogens coexist, the introduction of a virus can change the competition dynamics, impacting overall microbial diversity. In some cases, viruses might even confer advantages to their bacterial hosts, providing them with new traits through horizontal gene transfer, which can enhance resistance to antibiotics or other environmental stresses. The competition for resources among these pathogens often dictates their evolutionary trajectories as they adapt to one another. The arms race between viruses and bacteria intensifies, leading to rapid evolutionary changes. These interactions are not confined to single species. They extend across entire ecosystems, demonstrating that the health of one organism can drastically affect the others. Studying this interplay sheds light on the intricate balance that sustains life, emphasizing the interconnectedness of all living organisms, including those traditionally viewed as adversaries in the struggle for survival. Viruses serve as pivotal agents in horizontal gene transfer among eukaryotes, facilitating the exchange of genetic material between organisms that are not directly related. This process enhances genetic diversity, allowing for rapid adaptation to changing environments. Viruses can integrate their genetic material into the host's genome, a phenomenon that introduces new traits that may confer advantageous characteristics. This mechanism is observed in various eukaryotic lineages, where viral infections have led to significant evolutionary transitions. For instance, certain plants exhibit enhanced resistance to diseases through the acquisition of viral genes, which have become integral to their defense systems. In some cases, these viral sequences can be repurposed, leading to novel functions that were previously absent in the host organism. The implications extend beyond immediate survival advantages over evolutionary timescales. The infusion of viral genes into the gene pools of eukaryotic species can shape entire ecosystems. This genetic fluidity contributes to the resilience of these communities, enabling them to adapt to environmental pressures and shifts. The role of viruses in this context challenges traditional views of their impact, revealing them as key players in evolutionary dynamics. Their presence highlights the complexity of life's interdependencies, fostering an ecosystem where cooperation and competition coexist. Advancements in virus detection technologies play a crucial role in understanding these non-living entities. Innovations over recent decades have transformed the methods used to identify and characterize viral agents. Traditional approaches, such as cell culture techniques, relied on the ability of viruses to infect living cells, a process that can be slow and labor-intensive. The evolution of molecular biology, specifically polymerase chain reaction techniques, marked a significant shift. This method amplifies trace amounts of viral genetic material, allowing for rapid detection even when viruses are present in very low quantities. Next-generation sequencing technologies have further refined detection capabilities, enabling the analysis of complex viral genomes with unprecedented speed and accuracy. This technology allows for the identification of previously unknown viruses and provides insights into their evolutionary relationships. The integration of bioinformatics has also enhanced the interpretation of vast datasets generated from sequencing, making it possible to track viral mutations and outbreaks effectively. In addition, the development of point-of-care diagnostic tools has democratized virus detection, enabling rapid testing outside of laboratory settings. These innovations have proven essential during public health emergencies, as they facilitate timely responses to emerging threats. The continuing evolution of these detection technologies underscores their importance in monitoring and mitigating the impact of viral infections on global health. Viral ecology reveals remarkable adaptations that enable survival in extreme environments, illustrating the resilience of these non-living entities. In habitats characterized by extreme temperatures, acidity, or salinity, certain viruses exhibit unique structural features. These include modified protein coats and specialized enzymes that facilitate replication under harsh conditions. This adaptability not only highlights evolutionary innovation but also emphasizes the intricate relationships between viruses and their hosts, which may also be extremophiles. In thermal vents, for instance, viruses engage with archaea, tapping into their metabolic pathways for replication. The dynamic interplay between these microorganisms influences community structures and ecological functions. The presence of viruses can regulate population sizes, promoting genetic diversity and driving evolutionary changes among host organisms. Saline environments, such as salt flats, present a different set of challenges. Here, viruses adapt by developing mechanisms to stabilize their genetic material against desiccation. This includes the incorporation of protective proteins that shield their genomes, ensuring persistence in such inhospitable conditions. Acidic environments, like those found in certain hot springs, further illustrate viral adaptability. Specialized viral strains have evolved to thrive in low pH, maintaining their infectivity and contributing to the biogeochemical cycles of these regions. Such evolutionary adaptations underscore the complex role that viruses play in shaping life across diverse ecosystems. The influence of endogenous retroviruses on host evolution represents a remarkable interplay between non-living entities and living organisms. These retroviruses, integrated into the genomes of their hosts, become permanent residents, often exerting considerable effects on the genetic landscape. This integration can facilitate genetic diversity, as their sequences can be co-opted for various biological functions. Over time, these remnants contribute to the evolution of essential processes such as immune responses and placentation in mammals. In this context, the presence of these viral sequences has been linked to the development of critical pathways that help organisms adapt to their environments. Genetic material from endogenous retroviruses may serve as a reservoir for novel genes, providing the potential for innovation in response to changing conditions. The mechanisms through which these viruses exert influence are intricate. They can mediate gene expression and alter cellular functions, leading to phenotypic changes that may enhance survival and reproductive success. In certain species, the evolutionary trajectory has been significantly altered due to the insertion of viral elements, demonstrating how deeply intertwined these entities are with the host's evolutionary history. Across various species, this silent influence plays a crucial role in shaping not just individual organisms, but entire populations, illustrating a complex evolutionary narrative influenced by these non-living agents. The implications of this relationship continue to unfold, revealing layers of interaction that deepen our understanding of life itself. Case studies illustrate the complex interplay between viruses and their hosts throughout evolutionary history. In one notable instance, the co-evolution of viruses and mammals highlights adaptive strategies. Certain viruses, once harmful, have gradually become integrated into the genetic fabric of their hosts. These interactions can lead to the emergence of beneficial traits, enabling enhanced resistance against pathogens. The result is a delicate balance, where both entities influence each other's evolutionary pathways. Another example is found in plant virology. Specific viruses infecting crops have prompted defensive mechanisms in host plants, leading to the development of resistance genes. This ongoing arms race manifests in various ways, from altered growth patterns to the production of antiviral compounds. Such adaptations not only protect the host, but can also impact agricultural practices and food security. Research into these interactions reveals that viruses play a transformative role in shaping ecosystems. By influencing population dynamics and species distribution, they contribute to biodiversity. The evolutionary history shared between viruses and their hosts is a testament to resilience and adaptability. In aquatic environments, marine viruses heavily influence microbial communities. They regulate populations of phytoplankton and bacteria, thereby affecting nutrient cycling in oceans. Such interactions underscore the significance of viruses beyond mere pathogens, highlighting their integral role in sustaining life on Earth. Viruses contribute significantly to the intricate web of biogeochemical cycles, a testament to their pervasive influence on ecosystems found in diverse environments. They participate in the cycling of carbon, nitrogen, and phosphorus, acting as agents of microbial mortality. By infecting and lysing bacterial populations, they release organic materials back into the environment, facilitating nutrient recycling. This process ensures the continuous availability of essential elements vital for the survival of various life forms. In marine ecosystems, the impact of these entities is profound. Estimates suggest that viruses may lyse up to 30% of bacterial populations daily, generating dissolved organic matter that fuels primary producers. These interactions underpin the functioning of food webs, illustrating the delicate balance sustaining aquatic life. Similarly, terrestrial ecosystems experience the influence of viruses in soil microbial communities. By regulating microbial diversity, they maintain ecosystem health and resilience. Their role in nitrogen cycling is particularly noteworthy. Through the regulation of nitrogen-fixing bacteria, they influence the availability of this crucial nutrient for plants. Research into the specific mechanisms by which viruses drive these processes continues to reveal new insights. Understanding their functions within these cycles not only enhances comprehension of ecological dynamics, but also emphasizes their importance in the ongoing evolution of life across the planet. Research into these entities continues to evolve, revealing a vast landscape of potential applications that extend beyond their traditional roles. Innovative methodologies are emerging, allowing scientists to explore their genetic makeup with unprecedented precision. This insight opens doors to novel therapeutic avenues, including the development of targeted antiviral treatments and gene editing technologies. The implications of such advancements are profound. Harnessing viral mechanisms can lead to breakthroughs in combating chronic diseases, addressing global health crises, and improving vaccine efficacy. The exploration of engineered viruses for delivering therapeutic agents directly to infected cells represents a significant leap in medical science. In agriculture, viruses are being studied as biocontrol agents, providing environmentally friendly alternatives to chemical pesticides. Their natural ability to infect and eliminate harmful pests offers a sustainable approach to enhancing crop resilience. These ongoing investigations also delve into the role of viruses in shaping microbial communities within diverse ecosystems. By understanding these interactions, researchers can better predict how changes in environmental conditions may affect biodiversity and ecosystem health. Through comprehensive studies, the future of virus research promises to illuminate their multifaceted roles in both health and disease, paving the way for innovative solutions that could redefine our relationship with these complex entities. Ethical considerations in virus research and evolution are becoming increasingly significant as advancements unfold. The delicate balance between scientific progress and potential risks requires careful navigation. Researchers face challenging questions regarding the manipulation of viral genomes, especially when engineering strains for therapeutic purposes. The implications of creating more virulent or transmissible forms necessitate stringent oversight and ethical frameworks. In this domain, the potential for unintended consequences remains a constant concern. The introduction of modified organisms into natural ecosystems could disrupt established biological interactions, leading to unforeseen challenges. This reality compels the scientific community to adopt a precautionary approach, prioritizing biosafety and biosecurity measures. Public perception also shapes the discourse surrounding virus research. Transparency in communication fosters trust and understanding, allowing society to engage meaningfully with the implications of such studies. Ethical guidelines must evolve alongside scientific knowledge, ensuring that the pursuit of innovation does not compromise moral responsibilities. Collaboration between scientists, ethicists, and policymakers is vital for establishing robust frameworks that address these complex issues. Continuous dialogue about the societal impacts of virus evolution facilitates a responsible path forward. In the quest to harness the potential of these non-living entities, the importance of ethical stewardship cannot be overstated, guiding the exploration of their roles within the broader tapestry of life. The contributions of citizen science in virus evolution studies have emerged as a powerful force in understanding these complex entities. Individuals, often outside traditional scientific institutions, engage in data collection and analysis, enriching the overall comprehension of viral behaviors and mutations. This proactive involvement fosters a broader perspective on viral dynamics, providing insights that might otherwise remain hidden within the confines of specialized laboratories. In various regions, community-driven initiatives have led to the monitoring of viral infections, creating extensive databases that track changes over time. These grassroots efforts amplify the voice of the public, allowing for a diverse range of observations and experiences to inform scientific inquiry. Collaborative platforms enable shared access to findings, enhancing transparency and fostering a collective responsibility in addressing viral threats. Through educational outreach, participants gain vital knowledge about viral transmission and its implications for public health. This deepened awareness cultivates a culture of vigilance and preparedness, bridging the gap between researchers and communities. Notably, real-time data generated through citizen involvement can influence public health responses, shaping policies that reflect the evolving nature of viruses. As understanding advances, the integration of citizen science into the study of virus evolution continues to demonstrate the profound impact of collective engagement on scientific progress and public health outcomes. Historical pandemics have served as pivotal moments in shaping virus evolution and the trajectory of human health. Each outbreak has acted as a selective pressure, allowing certain viral strains to thrive while others diminish. The Black Death in the 14th century, for instance, altered not only the course of European society, but also the genetic landscape of pathogens. This period saw the emergence of more virulent strains that adapted to exploit human hosts, demonstrating an ability to mutate in response to changing environments. The influence of the Spanish flu in the early 20th century further emphasizes the dynamic interplay between viruses and their hosts. It highlighted the rapid mutation rates of influenza viruses, leading to the emergence of new variants that could evade host immunity. Such historical events underscore the necessity for vigilant surveillance and the continual adaptation of public health measures. In more recent times, the emergence of the human immunodeficiency virus has illustrated the complexities of viral evolution in the context of human behavior and societal structures. This virus has shown remarkable adaptability, leading to the development of diverse subtypes and resistance patterns. These evolutionary processes articulate a narrative of survival and adaptation, reflecting the urgent need for ongoing research and understanding in the face of an ever-changing viral landscape. The lessons learned from past pandemics remain vital in informing current and future health strategies. Transmission routes for viruses have evolved in intricate and often unexpected ways. These non-living entities adapt to their environments, developing mechanisms to ensure their survival and propagation. Airborne transmission, one of the most significant pathways, allows viruses to spread efficiently through respiratory droplets in crowded or enclosed spaces. This route becomes particularly effective, facilitating rapid outbreaks. Vectors also play a crucial role in virus dispersion. Insects, such as mosquitoes or ticks, serve as carriers, bridging the gap between wildlife and human populations. This relationship underscores the interconnectedness of ecosystems and the potential for zoonotic spillover, wherein viruses jump from animals to humans. Consequently, monitoring wildlife habitats becomes essential in predicting potential virus emergence. Contact transmission remains prevalent, especially in environments where hygiene practices are inadequate. Surface contamination, alongside person-to-person contact, fosters the persistence of viruses in communities. As societal behaviors shift, so too do the strategies viruses employ to spread. Understanding these routes is critical for developing interventions. Vaccination campaigns, public health advisories, and sanitation efforts are tailored to combat specific transmission methods. Continuous research into viral evolution and its implications informs not only immediate responses, but also long-term strategies for mitigating future health threats. The dynamic nature of these entities necessitates ongoing vigilance and adaptability in public health frameworks. The intricate relationship between viruses and the origin of life presents a compelling narrative within evolutionary biology. These non-living entities, while often causing disease, may also serve as pivotal players in the development of cellular life. The hypothesis suggests that early viral progenitors could have interacted with primitive cellular structures, facilitating genetic exchange and innovation. Such interactions are theorized to have contributed to the emergence of complex life forms. In this exploration, the concept of horizontal gene transfer becomes significant. Through this process, genetic material is shared between different organisms, allowing for rapid adaptation to environmental pressures. Viruses, acting as vectors, can enhance genetic diversity, driving evolutionary changes that shape entire ecosystems. Their role extends beyond mere parasites. They can be agents of evolutionary progress. The fossil record, while sparse concerning viruses, indicates their long-standing presence on Earth, potentially dating back to the dawn of life itself. This enduring association raises questions about the very definition of life and the boundaries that separate living organisms from non-living entities. In contemporary research, scientists continue to investigate the dual nature of viruses, recognizing their potential benefits alongside their pathogenic capabilities. Understanding these complex interactions not only informs public health strategies, but also enriches our comprehension of life's evolutionary pathways. This perspective underscores the interconnectedness of all biological entities within the vast web of existence. Viruses, often regarded as mere parasites of life, played a pivotal role in the emergence of eukaryotic cells. These complex organisms, which house their genetic material within a nucleus, owe part of their evolutionary journey to the interactions between viral entities and primitive prokaryotic cells. Through the process of horizontal gene transfer, genetic material from viruses integrated into host genomes, RNAs, facilitating novel functions and enhancing cellular complexity. In this region of cellular evolution, it is evident that viral components contributed to the development of essential cellular machinery. For instance, certain proteins derived from viral origins became critical in processes such as cellular signaling and the regulation of gene expression. This integration not only supported the survival of early eukaryotes, but also enabled them to thrive in diverse and challenging environments. The emergence of eukaryotic cells marked a significant evolutionary transition that allowed for greater adaptability and specialization. It is within this context that the role of viruses expands beyond mere replication. They act as agents of genetic innovation, driving the evolutionary processes through which more complex life forms emerged. These interactions illustrate the profound influence non-living entities exert on the living world. The legacy of viruses persists, shaping not just the course of individual species, but also the broader framework of life's diversity on Earth. Natural selection acts as a fundamental force driving the evolution of these non-living entities. In a variety of environments, they adapt through mechanisms that enable survival and replication. Specific mutations may confer advantages, allowing certain viral strains to thrive in the presence of host defenses. This advantageous shift often results in a rapid increase in prevalence, illustrating the intricate relationship between host organisms and their viral counterparts. The interaction between hosts and viruses can lead to a continuous cycle of evolution, with each influencing the other's genetic landscape. As host immune responses evolve, so too must the viruses, fostering a dynamic interplay that pushes the boundaries of genetic change. This ongoing process of adaptation contributes to the vast diversity observed within viral populations in environments marked by high mutation rates, such as RNA viruses. The potential for rapid evolution is amplified. This region demonstrates how genetic drift can lead to the emergence of novel strains, some of which may even cross species barriers. When this occurs, new viral lineages can emerge, presenting challenges for existing immune systems. Consequently, the impact of natural selection on viral diversity is profound, shaping not only the viruses themselves, but also the ecological and evolutionary dynamics of the ecosystems in which they reside. The interplay between these entities continues to influence life on Earth in ways that remain intricate and complex. Preparedness for viral pandemics shapes evolutionary pathways, influencing both microbial and host species. This region of study reveals how frequent interactions between viruses and their hosts can drive genetic adaptations. Over time, selective pressures imposed by viral infections encourage organisms to develop resistance strategies. This evolutionary arms race leads to the emergence of novel traits, enhancing survival in an ever-changing environment. Surveillance systems and vaccine development further contribute to the evolutionary landscape. The anticipation of potential outbreaks fosters innovation in genetic research, leading to advances in biotechnology. These efforts not only protect human health but also influence the evolutionary pressures faced by viruses themselves. As vaccines are deployed, only those variants capable of evading immune responses tend to thrive, altering the viral population dynamics. In addition, human behavior plays a pivotal role in this process. Urbanization, global travel, and agricultural practices create new environments for viral transmission. Such shifts can accelerate mutations and recombination events among viruses, fostering diversity and adaptability. Understanding these interactions unveils the complexities of coevolution, where both pathogens and hosts adapt in response to one another. Longitudinal studies provide valuable insights into how past pandemics have shaped genetic diversity in populations. This ongoing research informs strategies to mitigate future health crises, highlighting the importance of preparedness in the face of evolving threats. The intricate role of viruses within ecosystem dynamics reveals their duality as both agents of disease and participants in ecological balance. These non-living entities, despite their simplicity, hold substantial influence over population control among various species. By infecting hosts, they regulate biodiversity, ensuring that no single species dominates the environment, thus maintaining a delicate equilibrium. In aquatic systems, viruses contribute significantly to nutrient cycling. They infect and lyse bacteria, releasing organic matter back into the water. This process not only supports microbial communities, but also serves as a foundation for higher trophic levels, affecting the entire food web. The implications extend beyond immediate interactions. Long-term viral activity shapes genetic diversity among populations, influencing evolutionary pathways. Terrestrial ecosystems also demonstrate similar dynamics in soil environments. Viruses target specific microbial species, promoting the survival of more resilient organisms. This selective pressure fosters adaptability among microbial communities, which in turn enhances ecosystem resilience to climate fluctuations and anthropogenic disturbances. The interplay between viruses and their hosts is a fundamental aspect of life on Earth. Their presence is a reminder of the ongoing evolutionary arms race that shapes biological communities. Understanding these dynamics offers profound insights into the interconnectedness of life, emphasizing the need for comprehensive studies that unravel the complex relationships governing ecosystems. The future of viral vaccination strategies hinges on advancements in technology and a deeper comprehension of viral behavior. Emerging techniques such as messenger RNA technology demonstrate significant potential in crafting rapid responses to evolving viral threats. This region of research highlights the importance of adaptability, enabling the creation of vaccines that can be tailored to combat newly identified strains with unprecedented efficiency. Innovations in genomic sequencing further enhance the understanding of viral mutations, allowing for the monitoring of pathogens in real time. These insights facilitate the development of vaccines that not only address current outbreaks, but also anticipate future challenges. The integration of artificial intelligence into vaccine design represents another frontier in this endeavor. By employing machine learning algorithms, researchers can predict viral evolution and identify optimal vaccine targets. The prospect of universal vaccines, capable of providing broad protection against multiple strains, remains a focal point of ongoing studies. Such advancements could significantly reduce the burden of viral diseases worldwide. Collaborative efforts among nations, emphasizing shared data and resources, are crucial in this pursuit. This collective approach fosters resilience against emerging threats. This interplay between viruses and immune systems reveals a complex narrative of coevolution. Over millennia, these non-living entities have adapted to evade detection and destruction, while immune systems have simultaneously developed mechanisms for identifying and neutralizing them. This ongoing struggle is a testament to the relentless forces of natural selection, shaping both viral and host genomes. Viruses possess remarkable mutation rates, allowing them to swiftly alter their surface proteins and escape immune recognition. These alterations prompt a corresponding response from immune systems, which continually refine their strategies to recognize and combat these ever-evolving pathogens. This dynamic interaction leads to an intricate arms race, characterized by the emergence of viral variants and the diversification of immune responses. In this region of life, one can observe the fascinating phenomenon of cross-species transmission. Viruses can leap between hosts, introducing new genetic material into different populations. This not only influences the evolutionary trajectory of the viruses, but also has profound implications for the immune systems of the new hosts. The introduction of a novel pathogen can trigger widespread immune responses, resulting in adaptations that may persist for generations. Such evolutionary pressures have significant implications for biodiversity, impacting ecosystems and the interconnectedness of life. The resilience and adaptability witnessed in this relationship underscore the profound influence of viruses on the evolutionary landscape of all living organisms. Artificial intelligence has emerged as a pivotal tool in understanding virus evolution, providing unprecedented capabilities for data analysis and pattern recognition. Algorithms can sift through vast genomic databases, identifying mutations and tracing lineage with remarkable precision. This technology enables researchers to predict potential viral behavior, illuminating pathways that may lead to new traits or transmission methods. Machine learning models are particularly adept at discerning the subtle shifts in genetic sequences, allowing scientists to monitor viral evolution in real time. These systems can analyze historical data alongside current outbreaks, offering insights into how viruses adapt to host defenses or environmental changes. Such predictive capabilities are invaluable, equipping public health officials with the information necessary for timely interventions. In this realm, simulations generated by artificial intelligence facilitate experiments that would be impractical in a laboratory setting. By modeling viral interactions with their hosts or other environmental factors, it becomes possible to visualize scenarios that inform vaccine development and therapeutic strategies. The integration of AI into virology not only accelerates research, but also augments human understanding of complex biological systems. This convergence of technology and biology marks a significant advancement in the ongoing battle against viral diseases. It forms a foundation for future explorations, where the interplay between artificial intelligence and virus evolution may yield more effective responses to emergent threats. Genomic sequencing provides a crucial lens through which the intricate evolution of viruses can be examined. By deciphering the genetic material of these non-living entities, researchers can trace their lineage, revealing mutations that contribute to adaptability and resilience. This approach allows for a comprehensive understanding of how viruses interact with their hosts and the environments they inhabit. Through the analysis of genetic sequences, it becomes possible to identify patterns in transmission and virulence. The emergence of new strains can be monitored, offering insights into their potential impact on public health. By employing high-throughput sequencing techniques, scientists can compile vast datasets, enabling them to visualize the evolutionary pathways that viruses undertake in response to selective pressures. By illustrators, this method not only clarifies the mechanisms behind viral adaptation, but also aids in the development of targeted interventions. Vaccines can be designed with precise knowledge of viral variants, ensuring efficacy against evolving threats. The ability to predict shifts in viral behavior is enhanced, allowing for timely public health responses. The implications extend beyond immediate health concerns. Understanding viral evolution equips researchers with the tools to anticipate future outbreaks and to explore the broader ecological roles that these entities play in shaping biological diversity. Through genomic sequencing, the narrative of virus evolution unfolds, presenting both challenges and opportunities for science and society. Challenges in modeling viral evolution arise from the inherent complexity of these non-living entities. Their ability to mutate rapidly presents significant obstacles to accurate predictions. Each replication cycle can introduce genetic variations, leading to a mosaic of strains that complicates the understanding of evolutionary trajectories. This variability often results in unpredictable shifts in pathogenicity and transmission dynamics, making it difficult to establish reliable models. The influence of environmental factors adds another layer of complexity. Changes in host populations, climate conditions, and human behavior can alter viral evolution dramatically. These external pressures can drive selection in unexpected directions, further obscuring the relationship between virus and host. Data collection in diverse ecosystems becomes crucial, yet challenging, as sampling biases can skew perceptions of viral diversity and adaptability. Mathematical modeling techniques, while sophisticated, often struggle to accommodate the multifaceted nature of viral interactions. Simplifying assumptions may lead to gaps in understanding, leaving researchers to contend with uncertainty. The interplay between viruses and their hosts operates on different scales, from molecular interactions to global epidemiological patterns. Bridging these scales is essential for creating comprehensive models. Despite these hurdles, the pursuit of knowledge in viral evolution continues. Each breakthrough offers insights that refine existing theories and pave the way for future discoveries, enhancing the capacity to respond to emerging viral threats. The evolutionary landscape of life remains intricately intertwined with the presence of viruses. These non-living entities continue to play a significant role in shaping the genetic diversity of organisms. As they interact with their hosts, they facilitate horizontal gene transfer, introducing new genetic material that can lead to beneficial adaptations. This process illustrates the dynamic relationship between viruses and their hosts, highlighting them as agents of change within ecosystems. A deeper understanding of viral evolution is critical, particularly in the face of climate change and ecological shifts. Alterations in environmental conditions can lead to the emergence of new viral strains, which may possess unique capabilities to infect diverse species. This adaptability underscores the importance of ongoing research into viral genomics and their evolutionary mechanisms. Future studies will likely focus on the role of viruses in shaping not only the microbial world, but also higher organisms, including plants and animals. Investigations into their potential applications in biotechnology and medicine may offer innovative solutions for disease control and prevention. The continuous evolution of viruses ensures they remain relevant players in the story of life on Earth. As knowledge expands, the intricate balance between viruses and their hosts will reveal further complexities, emphasizing the need for vigilance and adaptability in our scientific approaches. Understanding this delicate interplay is essential for navigating the future of biological life.